Thank you, Juan, for the presentation, and thank you for the organizer for inviting me. As you can see from the title, this is something very different from what you heard until now, because we are talking about microbial toxin, bacterial toxin, botulinum neurotoxin, which is one of the main topic in the lab. We have many projects, but this is my first lab actually since my PhD. And so I will tell you about these toxins that are quite interesting because they can be represented as the Roman god Janus with double face, since they have a bad and good face. In fact, botulinum neurotoxins are the most potent toxins known, with lethal doses less than 1 nanogram per kilo. And for this reason, the American CDC included this toxin among the bioterrorism category A agents. They are the causative agents of botulism, which is a neuroparalytic syndrome characterized by generalized flaccid paralysis due to the blockade of the neurotransmission at the peripheral nervous system. On the other side, this toxin had a good face because they are safe and effective drug sold in many millions of doses per year and used for a plethora of therapeutic application. I will show you later. And again, the good face of this toxin is that they can be an important research tool in neurobiology. So based on this double face of toxins, my presentation outline would be the following. So at the beginning, I will show you the mechanism of action that makes this toxin the most toxic substance ever. And then the good face of toxin, so the therapeutic applications and the toxin as research tools, in particular in our lab experience. So let's start with the mechanism of action. As I told you, these toxins are bacterial toxin produced by gram-positive bacteria of the genus Clostridia, but not only Clostridium botulinum, also Butyricum, Barazzi, and Argentinesis, that are bacteria strictly anaerobic. So in the environment, they form spores that can germinate when the oxygen is low. Recently, thanks to the improvement of next generation sequencing, the interesting finding is that this gene, the gene encoding for botulinum toxins, bond-like genes, are also present outside the Clostridia genes. So in different bacteria, such as Enterococcus faecium, Bacilla oryza, Bacter piperi, and the list is continuously increasing. So it seems that this family of toxin is a large family that is continuously increasing. And classically, the Clostridial botulinum neurotoxins are classified in 7 different serotypes based on their immunological properties, activities. And again, systematic DNA sequencing of botulinum toxic genes and of Clostridium botulinum genomes demonstrated that Clostridia have considerable genetic heterogeneity, and many toxin variants named subtypes have been identified within each serotype. So we now have more than 40 different variants of botulinum neurotoxins. And this large number of toxin or variants brings along 2 major consequences, one bad and one good. The bad consequences is that the large number of botulinum neurotoxins poses a major problem to the current attempt to find efficient antidotes via antibodies and vaccine, because antibodies against one toxin can be less effective against another variant of the same serotype. And so this calls for identifying inhibitors that would prevent the neuroparalytic activity of these toxins in vivo by acting independently from their immunological properties. And this was the focus of our lab in the last few years. One of the focus in the last few years, as I will show you in a few minutes. The good consequence is that the novel variants provide a great potential of botulinum toxins, which might reveal novel therapeutic properties in terms of increased potency, specificity for different neurons, and also longer duration of therapeutic benefit. Despite the many botulinum neurotoxin, the many variants identified, they all have a conserved molecular architecture. They are formed by two chains, a heavy chain and a light chain, held together by a disulfide bond. And the crystal structure of the toxin reveal actually three domains: a binding domain, a translocation domain, and a catalytic domain, the L chain, which bind an atom of zinc, which is crucial for the catalytic activity of this subunit, which consists in a metalloproteolytic activity. So, as Mauricio Montal defined, this toxin is a marvel of protein design, which has been tailored around the essential feature of nerve terminal physiology, thus maximizing the toxicological performance and becoming the most poisonous toxin discovered so far. And this modular architecture is functional to the multi-step action of the toxin, which has the main site of action in the neuromuscular junction. So in the synapse between the motor nerve terminal and the muscle fiber. And here, this toxin acts by blocking the neuroexocytosis of the neurotransmitter containing synaptic vesicle. The neuroexocytosis is a very complex process which requires the implication of many different proteins, but the core of this process are three proteins, VAMP, SNAP25, and syntaxins, that upon the action potential, the increase of calcium inside the nerve terminal, form a heterotrimeric complex called snare complex, which is fundamental for the fusion of neurotransmitter containing synaptic vesicle with the presynaptic membrane, with a consequent release of neurotransmitter. So botulinum neurotoxins have a multi-step mechanism that culminates in the proteolysis of these three snare proteins, preventing the fusion of synaptic vesicle and so the neurotransmitter release. The mechanism of intoxication of the nerve terminal by botulinum neurotoxin is a multi-step mechanism which can be divided in 5 different steps. The first step is the binding, a double receptor binding of the of the binding domain with a polysialoganglioside and also a synaptic protein of the vesicle, which is available upon the fusion of synaptic vesicle with the plasma membrane. And this very high affinity binding is functional to the internalization of the toxin, mainly by the mechanism of recycling of synaptic vesicle. And some years ago, in collaboration with Jordi Molgo, by immunogold electron microscopy, we did the morphological demonstration that actually synaptic vesicles are the main Trojan horse for the internalization of the toxin into the nerve terminal. But I will show you that this is not the only way by that the toxin used to go into neurons. After the internalization, the toxin need to translocate from the organelles of internalization, and to translocate the catalytic subunit is fundamental, the reduction of the disulfide bond that keeps together the heavy and the light chain. And once the catalytic part is in the cytosol, then, as I told you, it proteolyzes the snare complex. So the disulfide bond reduction is crucial for the toxin activity. So a few years ago, especially Marco Pirazzini and Giulia Zanetti in the lab, decided to investigate which is the redox system that is responsible for the reduction of this disulfide bond. And to make a long story short, they found that thioredoxin 1, thioredoxin reductase is the redox system responsible for the reduction of the disulfide bond of all botulinum neurotoxin and for the related tetanus toxins as well. And so this system provided a target for the identification of potential pan inhibitors of botulinum neurotoxins. And indeed, they screen a lot of available inhibitors against these redox systems. And they found that especially this molecule here named epsilon, which is able to interact and block both the reductase and the thioredoxin, is indeed able to protect from neuroparalysis induced by all botulinum neurotoxins, serotype independently from the type of toxins, and also by tetanus toxin. So after decades of research and thanks to the effort of many laboratories worldwide, the understanding of molecular mechanism of nerve intoxication is presently very, very advanced. And this has significantly contributed to the safer and successful use of botulinum neurotoxin in human therapy. So the good side of the toxin. And this is the next point of my presentation. So botulinum toxin type A, in particular, this serotype A, is a formidable drug to treat human syndromes or conditions generated by hyperfunction of cholinergic peripheral nerve terminal, because these toxins, as I told you, are very neurospecific and very potent. So extremely low amount of toxin injected in precise sites is sufficient to block cholinergic terminals, which innervate muscle fiber, but also cholinergic terminal innervating the glands. This effect is reversible, but have a long duration of action. So in terms of therapeutic benefit, is quite important and quite long. In humans, it lasts 3-4 months in skeletal nerves, and even longer, more than 1 year, in autonomic nerve terminals. Another important properties is the limited diffusion from the site injection. This is very important that when you want to have a beneficial, a therapeutic benefit, you want to be precise in the effect and in the outcome. And then, very importantly, very low immunogenicity. This toxin is have a very low immunogenicity, and so very rarely we have a resistance of the patient treated with this toxin. So despite its extreme potency, botulinum toxin is a very safe drug. And the therapeutic uses of this toxin started in the late 70s by pioneering studies of Alan Scott, an ophthalmologist, who started to treat experimental strabismus in monkeys and then in patients. And from that studies in 1989, the Food and Drug Administration approved the use of this toxin for some focal dystonians, and then the list of human conditions that benefit from the use of botulinum neurotoxin is coming more and more longer and longer. And actually, they cover all the medical specialties, and in terms of neurology, for example, this pathology, blepharospasm, is a contracture of the eyelid, benefit from the injection of the toxin in the muscle of the eyelid, and the injection of very few amount of toxin give a benefit of many months. Again, hemispatial spasm, but also spasticity, tremor, and tics are pathological conditions that are treated by botulinum neurotoxin type A. And a serendipitous observation that this patient treated for blepharospasm or for hemifacial spasm have also distended wrinkles, give rise to the use of this toxin in aesthetic medicine. And at the beginning of this century, the cosmetic use was approved, and is really the real business of a company marketing this toxin, such as Botox. And as I told you before, the toxin block also the cholinergic terminals that innervate the glands, and so also hyperhidrosis, such as axillary and palmar hyperhidrosis, but also drooly, nasal allergy, and many other hypersecretory diseases benefit from the use of this toxin. But also in neurology, in gastroenterology, and very interestingly, more recently, the observation that these toxins are useful also in treating neuropathic or in general pain conditions. And again, a story of serendipitous, concern the treatment of chronic migraine, because some patients with chronic migraine that were treated for cosmetic use observed that the frequency of migraine attack was reduced by the use of toxin. And so in 2010, the use of botulinum toxin A was approved for chronic migraine headache. But what is the mechanism of botulinum toxin A in human therapy? As I told you, the main site is the neuromuscular junction. So it means that most of the toxin, after entering the motor nerve terminal, remain at the periphery. However, some therapeutic effects cannot be explained by the action only at the periphery. And so Matteo Caleo, at that time at the CNR in Pisa, decided to investigate if to look at for evidence of a central action of botulinum neurotoxin. And I take advantage of this presentation to give a tribute to the memory of Matteo Caleo, who unfortunately, very sadly, passed away a few months ago at the age of 51 only. And he was an outstanding scientist, a great friend, and great collaborators. And so we collaborated with Matteo by set up immunohistochemical test in order to be able to track the activity of the toxin after peripheral injection in order to see if the toxin can be act also at distant site. So for this purpose, we produced a polyclonal antibodies specific for the target SNAP25 of a proteolytic activity of botulinum toxin type A, an antibody which recognizes SNAP25 only after cleavage by the toxin. So by immunohistochemistry, we can detect the toxin activity by using this antibody. And for example, Matteo investigate many different circuits, such as the visual system, hippocampus, but also the motor system. And for example, upon injection of a whisker pad in mice or in rats, cleaved SNAP25, you can see the red spot here, was detected in the facial nucleus, so in the brainstem, in the central nervous system, confirming the hypothesis that the toxin can migrate from the periphery to the central nervous system. And so we were able to demonstrate that this toxin is transported by retrograde transport all along the axon, both in the motor system, but other groups then demonstrated also the retrograde transport also in sensory neurons. And this important observation is, the basis for the anti-nociceptive action of botulinum toxin type A. Therefore, the therapeutic efficacy of these neurotoxins is not only ascribable to the central nervous system plastic reorganization following the peripheral neuroparalysis, but depend also on trafficking of a catalytic active neurotoxin directly to the central nervous system. And the last part of my presentation, as I previously told you, is the use of this toxin as a research tools. So we use botulinum toxin, but also other toxins, as Marco Stazzi showed you this morning, as tools to study plasticity and regeneration capacity of the neuromuscular junction. And in fact, actually, speaking about specifically to botulinum neurotoxins, they cause a persistent functional denervation of the neuromuscular junction that triggers remodeling with sprout formation and new synaptic contents without any degeneration and no cell death, differently from other animal toxins, such as alpha-lactotoxin that mentioned Marco this morning. So Marco Pirazzini in the lab established a mouse model for studying NMJ plasticity by injecting the in-limb of mouse. These are transgenic mouse with green fluorescent Schwann cells. So they have the Schwann cell green. And he followed the remodeling consequent to the injection of the botulinum neurotoxin through the time from day 1 to 3 months after injection. And as you can see, after 2 weeks, but even earlier, sprouts start to be formed by the blocked neuromuscular junction, and they become longer and more complex with many new synaptic contacts after 3 months. And he observed, very interestingly, that sprouting is much more evident in slow motor neurons with respect to fast motor neurons which show little changes or any changes at all. So what are the molecular determinants of neuromuscular junctions plasticity, and how do we study them? Actually, we use the same approach that Marco Stazzi showed you this morning. So by transcriptomic, restricted to the neuromuscular junction affected by the toxin. So we collect the neuromuscular junction at different time after the injection of the toxin. We collected by laser capture microdissection, extract the RNA, sequence the RNA, and obtaining a transcriptomic profiling. So by computational biology, we select candidate hits such as growth factor or signaling axis that can be responsible for synaptic plasticity, and then we validate in vitro and in vivo. And this is the PhD program of Stefano Amoretti in our lab. So from this study that is just started, we expect to define the molecular determinants underlying nerve terminal remodeling, and to understand the molecular basis of the different capability of slow and fast muscle fibers to sustain nerve terminal remodeling. So this study, we think, will also contribute to understand the mechanism that they regulate the loss of plasticity in conditions such as aging or motor neuron diseases, which could be possibly considered and or targeted for therapeutic purpose. And I just finished my presentation with the citation of Claude Bernard that toxins can be used as means of destroying life or as agents for treating diseases. And I can add that toxins can also be used as a means to study physiological and pathophysiological processes in cells and tissue. And acknowledgement, I want to thank, of course, all the lab members, and first of all, Cesare Montecucco, that most of you know, is a mentor of all of us in the lab, is now retired from the academic commitment, but is still very, very active in the lab, and we are very happy for this. And then I want to thank all the people participating to the botulinum toxin project, and also the other lab member involved in our project and our collaborators. And of course, thank you for your attention.
So thank you also for keeping on time.
Thank you.
And give best regards to Cesare. Questions, comments? Please, Julia.
I have a very naive and ignorant question. So is it generally like when you get the botulinum toxin or the Clostridium is generally via injection, like via like food, or so it is ingested?
Yeah. Sorry, I didn't specify this point because of time, but botulism is mainly a foodborne disease. So the route of entry of the toxin is enteral, and but the action, as I told you, is neurological. So is the peripheral nervous system. So after the entry of a toxin, which is produced by bacteria in complex with our protein, in order to protect the toxin from the acid and harsh environment of the stomach. So the toxin arrives in the intestine and then is able, in a way that we are not well aware of, to pass through the intestine wall, and then through the circulation arrive, because of the very big neurotropism at the neuromuscular junction. Anyway, in our lab, we are also studying the effect of the toxin at the enteric nervous system. And Federico Fabris in the lab is dealing with this particular focus. And we have found that actually enteric nervous system is the first site of action of a toxin in a natural botulism. and the neurons in the enteric nervous system are able to uptake the toxin, and the toxin is able to act there. So it's not only the route of entry. The intestine seems to be the first site of action of the toxin.
I have a question.
Please.
Great talk, Ornella. I'm just wondering about the anti-nociceptive effect. You said it was because the toxin got retrogradely transported up the axons of the motor neurons, right?
Yeah.
So presumably blocking exocytosis centrally. But how does that stop nociceptive signals being sent to the brain unless it's also blocking exocytosis in the sensory neurons? I don't understand how that works.
Yeah, actually, there are a lot of work that has to be done to understand well the mechanism of action of a toxin, specifically in pain. For sure, there is a peripheral action of a toxin, because the toxin is able not only to block the release of acetylcholine, but also of nociceptive peptide at the sensory neurons, such as CGRP and substance P. But also, it seems that the retrograde transport to the dorsal horn of the spinal cord, for example, in neuropathic pain, is able to desensitize the system by blocking the release of nociceptive neurotransmitter also at the central nervous system. So it seems that the anti-nociceptive activity of the toxin is both peripheral and central as well.
Thank you.
Any more comment? If this is not the case, now, thank you again.
Thank you very much.
And